This method can help answer key questions about the direct role of tau in the nuclear compartment while excluding any potential cytoplasmic tau contamination. The main advantage of this technique is that it is broadly applicable to the study of the nuclear function of tau in other cell types and under different cellular conditions. Demonstrating the procedure will be Giacomo Siano, a postdoc from my laboratory.
Begin by plating 4 × 10 to the 5 SHSHY5Y human neuroblastoma cell line cells to be transfected with the empty control vector, 4 × 10 to the 5 cells to be transfected with untagged tau, 4 × 10 to the 5 cells to be transfected with tau NLS, and 4 × 10 to the 5 cells to be transfected with tau NES into individual wells of a 6-well plate. The day after plating, separately incubate 400 nanograms of DNA and the appropriate volume of cationic lipids into one tube of 250 microliters of reduced serum per well. After 5 minutes at room temperature, combine each vector with 1 volume of cationic lipid and incubate the DNA lipid complexes for 20 minutes at room temperature. At the end of the incubation, replace the supernatants in each well with 2 milliliters of fresh complete culture medium and transfect each well with the appropriate DNA lipid complex solution for an overnight incubation at 37 degrees Celsius. The next day, replace the supernatants with fresh differentiation medium. For Western blot analysis, wash the transfected and differentiated cells in each well with PBS and incubate the cells with 500 microliters of 0.1